Thank you, Prasanna, for the invitation, and I hope that I will be able to give a snapshot of what we are working on, the challenges that we are facing, particularly in the computational data analysis. And as you can, we will be seeing in the next hour or so that we really would like to get diverse views from people who are not in the field. And the reason being is that we are trying to reinvent the wheel in terms of computational aspects, data analytical aspects, and also plain just algorithmic approaches, which many of you might already know. So it will be really good to know if there are some new approaches that we can study the data and look at the patterns and look at the correlation. And one of the things that, as a student of science, we realize that the first thing, the correlation is, the causation is and correlation are not the same. So one of the things that we realize in biology is that there's so much of data we are generating, it's very easy to jump to conclusions. And we are always very, very cautious, particularly in the high throughput biology, that what you see is not necessarily what's actually happening in a cell system. So, what I thought I will do in the next hour or so is to give an example of what sort of high throughput data. I think Rohit, I missed his talk, but I'm sure that he had gone through some of the high throughput biology, the systems that's actually used to generate the data, and what kind of analytical parameters that people are using to analyze the data. But I'll go over one particular example from cancer, and in the Indian context, that what are the challenges that we are facing. But I'll also talk about some of the generic issues that the field is facing. So our lab is particularly involved in two different completely set of problems. So one is in the disease biology, which I'll be talking about today. What I'm not going to talk about today is something which is a very new initiative in the lab, but we haven't really done much, is in synthetic biology. And this synthetic biology is actually what we're trying to do is metabolic engineering of a particular compound from a neem plant, as many of you know. So again, I'll just briefly tell, because most of the talk is actually going to be in the disease biology area. As Indians, we all know about this plant, and we have used this plant some of the other points in our lifetime. So what we have done is that we have sequenced this plant. That means we understand the genetic code of this plant. We understand the expressed transcriptomic code of this plant from different organs. What we are trying to do is to put together the pathways that are responsible for production of the secondary metabolites. Secondary metabolites that are responsible to give pharmaceutical or agrochemical properties of this plant, and try to understand and engineer these metabolites in the lab by using standard synthetic biology mechanism. So in disease biology, we are studying cancer, and again in cancer I'll give you an example, specific example of a solid cancer of the mouth cavity, and I'll tell a little bit in detail why it is important to study the cancer of the mouth cavity in particular context to India. But before I start, just I must acknowledge these are the people who work in the lab, being an institute with a lot of floating students. So many students keep on going, and the funding agencies and some of the collaborators here and outside of India. I'll just take you back to biology 101, and again I apologize if you have already heard this. So as you know that a human can have trillions of different cells. So let's just consider a single cell, and this cell, all the genetic content is within the nucleus. Being eukaryotes or higher organisms, we have a cell wall, and being human, we have 22 different pairs of chromosome, including one sex chromosome, the X and Y chromosome in case of a man, and X and X chromosome in case of a woman. And if you just break open these chromosomes, these are nothing but just a string of 4-letter words, which we call as the DNA, along with the protein. So it's just beads on a string. So you have a long string. If I actually put all these strings in 23 different chromosome pairs together, that will be 3 billion letter long, or 3 into 10 to the power 9 letter long, along with different proteins. Okay, so we already know what these proteins are, and we already know now how these exact sequences read like in case of a human and many other organisms. Now this might actually sound very trivial now, but 10 years back it was not trivial at all. In fact, there was a lot of evidence in 1990 when the first human genome project got initiated, and the person who that time was selected to lead this project is none other than Gene Watson, the guy who actually discovered or co-discovered the double-stranded nature of the DNA. And during that time, and I'm talking about, of course, 14 years back or 15 years back, it was considered to be non-trivial. The reason it was non-trivial is during that time there was no tool or no instrument or no chemistry which was available to read 3 billion letters in a short stretch of time. But nonetheless, people did that, and during that time it cost nearly $3 billion US. And I'll show you some of the slides very soon that how there is a logarithmic decrease in the cost in this 14 years. And now you can do a single human genome sequence at a reasonable coverage, and I'll come back to the coverage, what I mean, near around $5,000 to $10,000, or maybe around 5 to 10 lakhs rupees, depending on how much you want to do. And the reason this is very important, when you keep hearing about genome sequence and money concerned, is actually not a static figure. The reason being is not a static figure. Ideally, in an ideal world, you would actually try to read this letter once, and that's about it. You will try to put them together once and for all, because your genome sequence is not going to change unless you have a disease, right? And that's exactly what we'll be talking about in the context of cancer. So, but the problem that happens is that the instrument that reads these sequences generates the errors, right? If there are 0 error, you can just read it once, and that's about it. That will be very cheap, inexpensive. But the problem is that these instruments have errors. So what you do is that you build a redundancy. That means you read the same damn sequence hundreds of times. Because these are random errors, the errors in that location is not going to be replicated in the second, third, fourth, and so on and so forth. So you build the model in such a way so you understand exactly where the errors are. More the sequence you do, lesser of an error there is. And obviously, mathematically, you can calculate what is the maximum depth you can go where the errors are going to be close to 0. Unlike physical sciences, biology is very inexact. Okay, you cannot make sure that errors are going to be 0. It will be close to 0. But the problem is this process is not inexpensive. More you sequence, more money you have to spend. So if you're spending 5 to 10 lakhs to do one, you can spend up to 50 lakhs if you want to do close to maybe 0.001% error. And we are not there yet, although the price is coming down. Okay. Now, having said that, this is the information flow inside a cell. Now, you're starting, of course, with something which is static, the DNA, and then you are going somewhere which is very dynamic. So anywhere between the RNA and protein is a very dynamic nature of the cell. And what I mean by dynamic, so if I take DNA from your skin cell, from your hair and from your toe, it should be identical. Okay? But if I take the transcript information from your hair, is going to be very different from that of your eye, because we know eye produces some of the proteins which are possible, or which make it possible for you to see. Hair is something which will have some of the protein which are structural, for example keratin. Okay? Now if I take the skin cell of one person who is white, Caucasian guy, and an Indian, well, Indian is a misnomer because they can be very dark to the very fair skin. If you take somebody who's a dark skin, obviously we know that dark skin produces more of a pigment called melanin. So any protein which is involved in the production of melanin, the corresponding RNA should be higher there. So what I'm trying to say is that from static to dynamic, all this information can actually be captured by using this instrument, what we call as next generation or the newer generation of DNA sequencers. Okay? Now, I talked about the human genome sequence project. I'll go just spend 5 minutes about how it was done about 15 years back. So the concept is actually pretty simple. You take the genome, and here as a long string, you break it, and this breaking actually happens either by force, enzyme, or very randomly. If I take a long string and I shake it vigorously, it's going to break. It's going to break randomly, and that's exactly what happens here. Once it's broken, then what we do is something called a cloning, what essentially means you take these short stretches of DNA and you wrap it around another long stretch of circular DNA molecule to make millions of copies of each circle. So imagine that this guy, there is a circle around it. The reason it's a circle because it's easy to manipulate and easy to amplify. We don't want a single copy, we want millions of copies. So how do you do that? You do that by process of cloning. You put that into a bacterial cell, the bacteria actually grows it for you, right? So now, if you go to the early genome centers, and the two initiatives that actually found successfully found out the human genome for the first time in 1991 is by the public initiative called NHGRI, where Francis Collins was the head, who is the head of NIH now. And one is a private initiative which was headed by Craig Venter, and then in Celera Genomics in Washington. they have completely two different methods. So this is the method which was taken by the public initiative. This is longer but more methodical to do the sequencing. So you have a single tube with a single type of molecule, and then you try to sequence that and then put those together. Time consuming. Now what the private initiative did, led by Craig Venter, they just didn't care. They said, we are not going to clone, that means we are not going to identify these short stretches. We just randomly fragment it and sequence it and let the algorithm by in silico mean, let these short stretches join together. Now there are obviously the algorithm actually has matured over the years. How people do that is essentially, if I have a stretch from A to A and a stretch from T to G, now you can see from this T to this A are identical, right? So if I put millions of copies together, anything which is identical is now you can make a single assembly from one end to the another end. Now there are advantages and disadvantages to this. The advantage is quick, less money consuming because you are skipping the middle step. The disadvantage is that you are as good as your assembly algorithm, right? So people have actually taken a combined approach, doing both later to finish the human genome. Now because of this advent of this technology, what has happened is, you can see the what I'm showing you here is how technology actually grown over the last many years. In the 2001 to 2010, and the graph you see there is actually something happened during the period of 2004, and this inflection point at 2004 happened because of the invention of newer generation of instrument, what we call next generation sequencer, right? And because of this next generation sequencer, you can see there is a huge jump in the output, and this output is kilobases, and also can be signified in terms of number of genome sequences deposited in the public repository. Now the field has actually grown so much, it has surpassed Moore's law. Now had we not been surpassing Moore's law, we would be growing as in the diagonal here, and as you can see this is the hard disk storage, typical Moore's law, and we were growing, this is the pre-next generation sequencing era, we are growing way slower than what actually we are growing right now, which is the post next generation era. Essentially, going back to Gordon Moore when he was telling about the doubling of the transistor in 18 months, we way surpassed that time. And in fact, this whole field only started post 2006 because the first commercial instrument to produce the data was introduced in 2006. So we are not even 10 years old, and the amount of data we have generated are way more than even exabytes. There are actually centers, individual centers who produced more than exabyte of data. In fact, it's strange that 5 years back I even didn't know what is an exabyte. I had to look at the Wikipedia to see how many zeros are there, because we hardly dealt with anything which is more than a gigabyte or terabytes. And now there are individual genome centers whose IT expenses are in the range of $300 to $500 million. So that has challenges of storage, challenges of compression, challenge of decompression, challenge of analysis, sharing. And imagine if you are collaborating with 5 different international centers, how do you collaborate? How do you send the data? Cloud is completely useless for Indian context because pushing the data is a big problem. How do I push 1 terabyte of data through a wire? I'm paying 3 lakhs rupees a year just for 3 Mbps speed. So cloud it can only be good if I have the data there. But how do I send the data? The only way, and right now, is physically shipping the hard disk. That is the only way. You may not like it, but that's the only way right now, because we don't have the speed. No matter what people say, we don't have the speed. It breaks 100 times. So we can't even download a 2 GB file. Forget about TB, terabytes of files. So those are the practical challenges. So how do you deal with that?
By the way,
yeah.
Which is the biggest, how big is the one in Shenzhen?
The Shenzhens are right now reaching about 2 exabytes.
And is it the world's second largest, or I don't know?
Yeah, it's either they are going back and forth. So there are 5 large centers, one in Shenzhen, one in Washu in St. Louis, one in Baylor, Houston, and Broad in Cambridge. These are the 4 largest. They are all in the similar range. Very large. I mean, we are nowhere close to that. So anyway, so this is basically the same slide tells you that because of this inflection point of the data, you actually have a huge number of genomes sequenced and deposited in the public database. In fact, this particular slide could be very dated because it's 2009. In this field, 2009 is extremely dated, it's dinosaur. Things are changing every 2 months. And this is another problem because in the government grant, as you know, you write a grant, by the time government gives you money, things have changed. Then you say, I don't want that instrument anymore. So you wrote about this. I said, yeah, the committee met 6 months back, things have changed. So this is the reality in this field. So when you write the grant, you have to actually foresee what's happening in a year's time and write a grant accordingly, which is very difficult here. And these are the type of data, amount of data we are talking about. So in a single instrument, and again, there are multiple types of instruments, so I just want to simplify it. In a single high throughput instrument, we are talking about 30 to 60 terabytes of space, minimum space that you would need to deal with. Now, it's not unusual to have small genome centers to have multiple these instruments. So I'm talking about you need to, and this is per year, okay? So if you have 15 students, if you have 2 or 3 of these instruments, you need to have at least 300 to 400 terabytes of storage. Now, the problem is storage is cheap, but good storage is not cheap. If you are thinking about RAID 6, NFS kind of storage is very expensive. So now this is another problem. Now, who do you write the grant for storage? DBT says it's not our job. DIT says you are in biology, it's not our job. So this is the practical problem. So who understands biology and data both? So this is the practical problem that we all are facing, and in fact it's not specific to us, but the field is evolving, so we'll all have to deal with it together. But anyway, what are people doing? And we are also doing similar things. These are not going to be new to you guys, this audience, so some of these things we are also trying. I'll show you one example where we have used the GPU accelerator to reprogram one of the commonly used algorithms. And okay, so let's talk about an example, what I'm going to talk today on cancer. Essentially, you take tissues, there is a black box for you, you don't know anything what's going on, and you get an answer. So you already seen, and I told a little bit about the intensity of the data, the complexity of the data, the size of the data that one has to deal with, but you haven't even begun to understand the complexity of biology. Now, if you think the data is complex, welcome to biology, where everything is damn complex. So now, if you just add that on the top, this complexity is so high, many times you think that you have discovered something, but you are nowhere close to the end. Okay, and I'm going to show you one story. Now, cancer. Why study cancer in particular relationship with genetic changes? Now, I'll show you one of the very dramatic figure. I don't know whether because the light you may or may not be able to see. So what I'm showing you is picture of 2 patients before and
you said we have so much data. What is the quality of the data in that? I mean, okay, you can have a petabyte also, but often to make a petabyte of data is meaningless without a model.
So what do you mean by quality? Define quality.
The quality is definitely, well, okay, I suppose if you have you have sequenced, let us say, 1 trillion base pairs. What's the percentage of base pairs which could be in error? There's probably I don't know, there's some percentage could be in error. The bigger thing is what information have we been able to, do we have at least a semi-automated procedure to try to look at the data and say this probably came from this kind of organism, and the particular physiology is, okay, let me be very, very crude, healthy or not healthy. That's all. Now I'm not even saying to classify which disease or so, healthy or not healthy.
You know, the problem is that it's a very qualitative assessment. What do you consider healthy? None of us in this room is healthy.
Okay, all right, so you can define something.
No, I mean, this problem is that when you say asymptomatic, unhealthy, here is the problem though. Anybody who is above the age of 40 is not healthy. You have some problem, which is not serious enough.
Okay.
So, that is actually an important problem, because, a lot of people, what happens is that when we take somebody as control, it's easy to say about cancer and non-cancer. But is it easy to say a gastric related problem than a complete healthy child? It's not. So your question is that can we at a sequence level distinguish between a healthy and non-healthy? The answer is yes. That's what I'm going to show you in cancer, but it depends on the disease. So for a disease which is as complex as multifactorial as diabetes, maybe T2D type diabetes type 2, it's not that easy. So in a pure genetic disease, it's easier in cancer. But you can't just say that one size fits all. I'll just make one particular diagnostic platform where I'll distinguish between healthy and non-healthy, because it's not the shade, just the black and white, it's the shade of gray in between.
Okay.
And that's what makes it complex.
So can we say something like prone to flu? Do we have a genetic means of saying prone to influenza?
So a lot of work actually people have done in influenza. As far as I know, there is no loci in the genome which determines. So far, I am not aware of whether anybody has done a large-scale association studies on influenza, which is the only one which will answer that question. But as far as I know, there is no loci yet discovered in a large study which will actually pinpoint a particular locus to that of the susceptibility of flu. Not. But people have done that study in many other diseases. For example, I told you about the diabetes. People have done that with 100,000 individuals. But again, the association between a genetic locus and the susceptibility is actually very poor. The reason is that these are multifactorial diseases. It's not genetic component only adds certain percentage to the susceptibility. Okay, so here what I'm showing you is just basically telling why study genetic changes in the context of cancer. So it's no brainer to you if you just see the left and the right, one patient here, one patient here. What I'm showing you the patient, two patients who actually had skin cancer, is called a melanoma, and they are treated with a specific drug. Now, the reason I'm showing you this particular two different patients is the drug against which these patients were treated were very specific against a kinase, and this bemurafenib is actually specific to something called a gene BRAF. Okay, that BRAF has a mutation, okay, at a very particular location. In fact, the location is 600. Is a valine to glutamic acid change, so V600E change. So imagine I talked about the 3 billion letter long. In this 3 billion letter, a single letter change actually makes you susceptible. Either you are going to be acting positively for this drug or not, right? So if imagine if I have this disease, I would like to know whether I have that change before I take that drug, because that's going to be a do or die situation for me. And hence, in cancers, and this is obviously an extreme example, in cancers it's extremely important to know the genetic components that are involved in the cancer. Now you must distinguish that I am only today going to talk about non-hereditary cancers, not hereditary cancer. For example, breast and ovarian cancer, there are certain forms of breast and ovarian cancers who are hereditary. Indians are not necessarily highly susceptible to this, but certain cases, for example, the white Jews who migrated from Europe post World War II to US, they are called Ashkenazi Jews. They are highly prone, because of the hereditary carrying one particular allele of BRCA gene, they're highly prone to the breast and ovarian cancer, right? So I'm not talking about hereditary cancer. I'm talking about the cancers that we don't inherit from our parents. These are the cancers that the genetic changes happen after we inherit or acquire bad habits like smoking, drinking, and etc. Right? Now, in the context of India, what I'm showing you here, I'm showing you a graph which has both incidence and mortality. Incidence is simple, how many cases we find. Mortality is how many cases out of that are dead, right? High incidence doesn't necessarily mean mortality. Now here if you see, here is India and here are the other countries that matters to us. If you see the total cumulative additive numbers, actually very low in India. But if I show you the incidence, mortality as a percentage of incidence is one of the highest. The reason being is most people who have cancer in our country show up in the clinic at a very later stage, right? But in case of US, because of the very rigorous and large scale screening programs, they actually find it much earlier. Now, if I show you the comparative cases for a cancer in the oral cavity, now if you see the US and Japan, two Western countries, one in Asia, one in the northern hemisphere of America. Now I'm showing you the top 15 cancers, both in blue incidence and red in mortality. The cancer of the oral cavity doesn't even appear in the top 15. But if you look at the case in India, it's right there in the third most cancer in terms of both incidence and mortality. Hence telling that studying the cancer of the oral cavity is very important in the context of our country. Now, there is a huge disparity in outcome. If you just compare the oral cavity cancers between one particular city, here the statistics come from hospitals in Mumbai, and the entire database of the US, which is the SEER database. Now there is a whopping 20% difference between two different populations, okay, on survival. Now that is primarily due to about a quarter of our patients, as I mentioned earlier, show up in the clinic at a very later stage in comparison to those in the US. So essentially this is the localized cancer, that means the cancer which has not spread to different parts of the country, body, and the locoregional cancers which have spread. And in India, most of the people actually show up at a much later stage. So we have one distinct problem here, what we call as downsizing. That means we'd like to see more and more patients at an earlier stage, because that has an impact on survival ultimately. Now coming back to what we are studying, so this is just a lateral view of the head and neck region, and as you can see, there are multiple different organs, and we are going to study one by one, but right now I'm going to show you the example in particular connection with the oral cavity, and that too specifically on oral tongue. This is how it looks like. So this is the oral part of the tongue, and we are not going to study the rest of these other people who are studying. We are only studying a part of the oral tongue, and I'll show you the reason why we are interested in oral tongue. Maybe I'll just show you that slide first before coming back. On the top, I am showing you the overall incidence of the cancer in the oral cavity, including oral tongue. As you can see, this is sorry, these letters are pretty small. This is a database from Chennai, which suggests that the overall incidence of the cancer in the oral cavity is coming down, but that of oral tongue is actually going up. But if I substratify the patients further, of only those people who are below 45 years of age, it's even drastically going up, right? So why the cancer of one particular organ is drastically going up actually requires studying. In fact, this is the database from the US, which essentially says the same story. This is the overall incidence of all the oral cavity except tonsil and tongue. This is the overall incidence in oral tongue, again suggesting that oral tongue incidences among young people is going up. What we are studying right now, if you can see, this particular patient has a white patch, and this white patch on this side is actually developing tumor. Now about 25 to 40% of the people who develop white patch in the tongue develop tumor, not all, okay? So what we are interested in finding out, what is that in these patches which make it to develop into cancer, number one. Number two, what is that in non-white patches that also develop into cancer? Once we find out the genetic or the transcriptomic or epigenetic signatures, we would be able to detect them early. The whole idea is to detect them early, number one. Number two, to make sure that the treatment that is given to in the hospital to those patients actually is working. Because you'll be shocked to know about less than 20% of the treatment given to the cancer patients are ineffective. And that is the reason we are in the era of personalized genomics or personalized medicine. We want to make sure that based on the genomic profile, we actually identify or stratify those patients and only give to those patients the targeted drugs, for example, where it is going to act. Okay, I already showed you this. This is a slide I can skip. It's more on biology, that's why exactly we're interested in tongue versus the buccal. And this is how the patients actually look like. This is the white patch. develops into tumor. This is how it looks like if you just take a cross section and look it under the microscope, a complete hypoplastic tissue to that of the cancer tissue. Now take a step back right now, what I have shown you so far. So I've shown you the challenges of getting a large amount of data out of these instruments, and what is the extent and amount of data that we are generating, or centers like ours are generating. Then I switched back and talked to a little bit about the cancer and cancer of the oral cavity. Now let's go back to the data and try to understand that what exactly is the problem in a small investigator driven lab, for example, people in the context of India in general, but also in the rest of the world are facing. Now when these tools were made available commercially in 2006, it actually created a very interesting dynamic among the CS people and the biology people in the US. So it was a perfect opportunity for some of the young students in CS to try and write some of the tools, algorithms, try different combinations using the biology data. Now the problem is that biologists are traditionally not trained in analytical techniques. They're not comfortable in analytical techniques. They are not comfortable at all doing anything to do with computers except the menu driven or GUI driven things. And that started changing in 2006, 2007. And most of the labs actually started to struggle to deal with this data, what to do. So they were scrambling to get grad students in CS, but coming back to the biology. Now in India, of course, that problem is acute even today. You don't have enough number of people who are trained to look at data and are savvy analytically to look at the data and understand biology to do those together. So we thought that maybe one of the things that we want to do as a small lab is to come up with a combination of tools which are already existing in an open source database. Mostly students and postdocs and scientists have published, and find out what combination of the tool and why works best for the cancer analysis, particularly cancer exome analysis. And when I say exome analysis, I'm talking about the analysis of about 2% of the genome, because it's much easier to deal with this smaller database. And that's exactly what we did, and we published a paper, but I'll show you that this copy number analysis first. So one of the things that we tried to do here, this is basically a genome in chromosome 11. Okay? Now a single dot here represents a signal intensity at one particular location of the genome. Now it's no brainer for you to see that actually there is an increased signal from this stretch and this stretch. So think about it, that your chromosome has two copies, one from your mom and one from your dad. And if there is a gene, a stretch of this ATGC letter, which is repeated 10 times, I should get a signal proportional to the number, right? Now here, obviously the number of copies which is present between here to here is way higher than the normal copy, and again between here to here is higher than the first state. Now by looking at an absolute copy number, we can actually say that this is present at a normal copy number, which is diploid, because I have two copies. This is present at one allele is now amplified instead of having two alleles amplified, and at this particular stretch I have both the alleles amplified, right? Now this is a state we describe an association with cancer, and particularly oral cancer.
Can you repeat what is that second graph saying?
This is
How
just
is it different from the first graph?
So this is a subset of this graph. I am showing you basically three different stages. This is nothing but just a quantitative measurements of a stretch of chromosome. So if you take a chromosome, and if I have this guy repeated 10 times, if I get a quantitative signal from this, and if it's repeated 10 times, it will be 10x, right? So this is my baseline, which is 2x, because human being is diploid. Now this is 3x and this is 4x. And the difference between what I'm showing you is, it is possible by this technique to distinguish a single copy amplification from a two copy amplification. Out of these two copies that you inherit, one from paternal, one from maternal sides, here in this case a single copy is actually amplified. In this case both copies are amplified. So by using sensitive techniques like sequencing, you can do this. Now, in this particular case, this may not be diagnostics, but in many cases, copy number variation can actually be diagnostics. In fact, in many mental disorders, particularly in autism, people have actually found that it's very important that CNVs plays very important role, the copy number variation. Now, this is something that we do for the cancer sample. So I'm showing you multiple different samples on this axis here, and I'm showing you these are individual patients, okay, and I'm showing you here the number of different synonymous to non-synonymous single nucleotide variants. Again, these are different terms. Essentially what it means is whether if A is changed to T, does it actually change the amino acid or not? Because ultimately what matters is a change in protein. So if there is no change in protein, we call it as a synonymous change. If there is a change in protein or there is a stop codon, that means the protein is abruptly stopped, we call it as a non-synonymous change. So essentially I'm just showing you how many are disruptive changes versus how many are non-disruptive changes, and in different patients. And these are just the color coding for showing that whether these particular patients, where there is a recurrence, that means the cancer has actually come back post-surgery and radiotherapy and chemotherapy. In these cases, it has not recurrent, so the blue is no recurrence, red is recurrence, and then in blue, the yellow cases where the patient has actually died. So essentially what we are trying to do is get this information. Now if you step back one, all these cancer tissues we are getting, we're trying to understand their sequences. We're trying to understand how the sequence of a cancer tissue different from a normal tissue, okay? And if we find a difference, can we correlate that difference with death, recurrence, activity of a particular drug, right? That's exactly what we are doing. Now we are also trying to ask many different fundamental questions. For example, what is the mutational burden in a patient? For example, how many mutations are there in the genome? So if you take a normal genome, if you take genome of two individuals who are sitting next to each other, they will differ in a letter in every 500 to 1000 letters. Now those are not called mutations, those are called SNPs, or single nucleotide polymorphism, but they are benign changes. Just because you have different SNP from him, you don't have a disease. The reason being is that change in DNA is not associated every time with a disease, and the moment it is associated with a disease at a particular frequency, we call it as a mutation, and those are deleterious mutations or the bad mutations. And here our interest is not to find the difference in DNA that is trivial, but to find the change in DNA which cause disease and can be associated with progression, efficacy of a drug, or recurrence of cancer, right? And again, these are the same different parameters that I showed before, and we're just trying to map what kind of changes are happening in these patients, whether it's a G2A or G2C. Remember, there are 4 different nucleotide, A, T, G, C, or G2T. The reason we are doing that, because there are some studies which actually find that in case of a Tobago-related cancer, a transversion, particularly a guanine to thymine transversion, which is G2T transversion, is pretty common. And we didn't actually find that a strong correlation in our patients. And finally, we want to show a genome-wide data. Again, this doesn't really make any sense to a layman. What I'm showing you is from chromosome 1, and this is a male, all the way to chromosome X and Y, what are the different changes and where are they? What are the different single nucleotide changes in pink? What are the different insertions and deletions in yellow? And what are the different copy number changes in brown? Now you can see that in a single individual, there are so many different changes. And I am not even showing you all changes, including transcription, methylation, structural variation. Only at these 3 levels also there are so many changes, essentially telling you that the cancer genome is actually extremely changed in comparison to a normal genome. Now our job is to find out how much of that change is actually associated with the disease and how much of that change has no effect. Now, having said that, the challenge in the field is actually the number, and I'm showing you the next slide. Where is the number challenge coming from? So this is pretty self-explanatory. What I have done here, I've done a power calculation, okay? How many patients that one should study to catch or to capture all the changes at 1% frequency, okay, in our cancer, considering a fixed rate of mutation. So if I know that this institution produces one brilliant student 1 in 10 years and has an intake capacity of 100, how many and has a faculty strength of 20, and out of which probably 50% of the faculty are very good. Now how many years will it take with the retention rate to produce a genius? Now here, and that is close to 10,000, right? I have to take 10,000 samples to catch changes at frequency 1%. Now of course I can catch which a much lower number if I have more common variants, but some of those variants are not very common, they're extremely rare, and to catch those rare variants I have to actually study 10,000 patients. Now including the cost of analysis, people, sequencing, at today's rate, if I do it at the whole genome, it will cost nearly 200 crores. Now you got the answer, it's impossible for us to do. Even at a coding sequence level, which is 2% of the genome, if I want to do that, that will take me 20 crores. And the rate at which the government gives us money, I mean, we'll probably never get that money. So now the question is that under the current circumstances, most of our studies are going to be underpowered. So having said that, there are international efforts that are going on in all cancers, not just in oral cavity cancers, to capture all those small changes at a very, very small frequency in most of the cancers. And people have already actually produced data on more than 3,000 to 4,000 samples across the world in consortiums. In genome science, most of the large studies actually are done in consortium mode. So it's very common for an investigator to be working in consortium, not alone, because it's just impossible. It's very large amount of money is required. And it's not uncommon to have people in the lab, 100, 200 people. Half of them are doing analysis, half of them are probably producing the data. But that kind of collaborative consortium culture is unheard of in our country. People don't get along within the same institute, forget about across the institute. So it's just impossible. There's a big, and that's the truth in every institute. That's the case. So there's just big ego, big things, and then there are also big guys, small guys. There are issues. So now, how do we change to produce those data in a large consortium mode, which will be beneficial to the country, is actually a challenge. Okay. In addition to that, we also have additional challenges of cancer. These are challenges which are biology challenges. Okay. The reason is cancer is extremely heterogeneous. What do I mean by heterogeneous? If I take a bunch of tissue in the operating room, not all cells in those cancer tissues are same. Ultimately, I'm getting a cumulative average signal because I'm just crushing it and getting a signal. Now, how do I know the signal that I'm getting is actually representative of the cancer cell? The answer is we don't. What people are trying to do, they're trying to move into the single cell level. And as you can imagine, then just make 1 log higher cost. Extremely challenging technically, in addition to the cost. But that's what the field is moving now. Now, I already mentioned to you that just because I found a change doesn't necessarily mean that's the change which is causing cancer. Correlation and causation. Now, how do I find relevance of these thousands of different variants? How do I actually design functional studies to say that in fact the change that I have detected is indeed the change that causes cancer? So these are some of the biology related issues that one has to deal with studying genetics of cancer. In addition to that, there are data related challenges. I talked about few of them. Obviously, huge amount of data sharing, storing, archiving, but also analysis and visualization. You heard perhaps this morning from Rohit about some of the challenges and how to deal with that, and I think Durai mentioned about the proteins, which has even more complex than dealing with DNA or the transcripts. Now, how do we visualize this? How do I visualize changes in the genome which are megabases or even more apart from each other. How do I know that a chromosome 1 protein is interacting with a chromosome 22 protein? How do I visualize it? Biologists work best with visual things. They can work with abstract things. Now, how do I visualize this? The people are thinking about multiple ways of representing the genome, but we don't have a perfect solution yet, and that's one of the challenges. Now, obviously, the end point is, how do I find? We have time?
Yeah, we have plenty of time.
Okay, good. So, how do I find biologically relevant answers from this data? Yeah.
Actually, it's precisely what a question I want to ask. We have this famous Smith Waterman and all these algorithms about sequencing, and they basically look at the score. And the score is accumulated as token by token as you go along. And it's the probably textbook example of dynamic programming algorithm. But how it's entirely possible that a sum of scores is really not biologically relevant, and you just because there can be correlations between widely separated regions which could be causing some particular physiological feature to appear. So, of course, almost everybody just seems to maximize the score and just find the one which aligns the sequences best and so on according to the score. How would I'm sure people have thought about this, whether score maximization is really biologically relevant, or what is sort of the consensus on this? Because exactly what you said, if it turns out there are some global matching issues, the algorithm's complexity will just dramatically go up if you simply cannot add the scores.
So people are aware of it, and they actually have devised some ways. So it entirely depends on the context of the genome. For example, if a genome is known to be highly repetitive, so if I know a string is repeated X number of times, then I know that the score will be much higher in all those places. So for example, one of the common things people do before calling variant, after doing Smith-Waterman or Burrows-Wheeler kind of transformation, is to do local realignment. So right now many algorithms actually don't throw away the strings which has the second best score.
Oh, they don't?
No, they keep that. So you can keep that in the table and take into consideration in step 2. So right now actually that problem is not that severe. The reason being is that not only you can keep the score, but also finally when you find the variant of your choice biologically relevant, you can always go back to that particular string to see what's the score of number 2 guy, what's the score of number 3. So you can actually design. People do local realignment, but people actually do in fact increase the number of gaps and mismatches to find. So that's not that much of a problem. And with string length getting bigger, longer, that problem is even getting diminished day by day.
Okay, that is, it's not very common, you say, but the question is, there's these things that small change differences in some places can be extremely critical for the organism.
Yes.
So the question is, if you could have a score which is high but which is wrong in those places.
It happens all the time, and that is the reason people always have to go back and validate. So if I have a place which I think is relevant for cancer, I can't just publish just because of one experiment. I'll have to go and take another 50 different patients and find out how relevant that is. I have to design functional studies that if there is a change which I think is linked with cancer, is it really linked with cancer? So I have to do downstream biological experiments to prove that. So people do that routinely. It's not, it's impossible to design an algorithm which is going to be perfect. That means there is one score on one strings. So the only thing is that with the assays and chemistry getting better every day, string length is getting longer every day, your chance of getting a perfect score for your string is increasing day by day, but it's not perfect yet. Okay, so those are the some of the data challenge, and I think this is pretty obvious to all of you. I am sure that what exactly we need to deal with this environment in terms of the data, and most of the people are dealing with it. You have another question? Yeah.
Yeah, I have, but I
Okay. So what I'm going to show you a few examples of being a small lab, that what we are trying to do to, and these are primarily the projects as I mentioned. You can imagine how small and insignificant we are in comparison to the large labs in the world in this field. And these are primarily the projects which are designed for students to, get their hands dirty in the field. One of the things that we faced when we started about 3.5 years back, is for any algorithm to test and to write any tool, we need to have a simulator. We don't want to deal with the real data. Real data is not good because it's messy, it's noisy. We didn't have a simulator which actually can do multiple things. We did have a simulator which can do one particular variation simulation. So, then one of our scientists, actually what he did is he tried to design a tool which can simulate multiple different variation as well as generated read, but by taking the error profile of the instrument in question. So that means you simulate based on the actual errors which are the instrument generating. So it's closer to the situation in a real situation. But at the same time, the good thing about it is that because you have simulated it, you exactly know how many variations are there. So any tool or algorithm you write post that step, you can actually test it. This is one of the things we published. So basically, the second thing we try to do, and again, as you can see, these are very standard thing, might sound very standard for you guys, but it's not standard in a biology lab. We thought, can we take an accelerator, so just a bunch of GPUs, and see that can we just fasten a well-known algorithm, and in this case, of course, is the Borel-Wheeler's transformation. And in fact, we did that, and we can show you that in a serial code in comparison to the parallel code, there's a huge difference. But this probably is not the best, because we don't know how to write CUDA codes as good as probably other people. But we just had the first cut, and then we compared that with some of the other parallel algorithms, and we find that ours is not that bad. It's not much better, but it's okay. So perhaps these are some of the things that needs future refinement. In fact, a lot of people are now writing parallel codes, both in using the Intel mic and NVIDIA's CUDA, and trying to implement it for different steps. The problem here is having just one tool implemented or with accelerators is not good enough. You need to have from end to end with the accelerators, because if I'm going back to the same X number of clusters after 5 minutes, it doesn't really help me. So these are academic projects. It actually doesn't help. What helps, or what will help, is if I take the raw sequence from the instrument and I have 15 algorithms all parallelized, and I push a button and I get the results at the end, and that will have clinical relevance. This doesn't have clinical relevance. Remember, in the clinical atmosphere, you have zero chance of making a mistake, because a guy is going to diagnose or prescribe something based on that single A to T change. So your chance of making a mistake is minimum. Now, how do you do that? And that's the challenge. Now, that challenge is here. It doesn't even end here, and we actually have in a very nascent stage how to deal with that. Some of the challenges that I have written here is primarily comes from the US environment. Now, we don't have any standards. I think as a race is probably something to do with genotype. We are great in developing new methods, new things. We're just lousy in following standard things. And that is a necessity in a clinical environment. That means don't fiddle with, don't change. If it works, it's okay. But you always have some students say, no, I can do it better. No, you don't need to do it better. So you have to lock it. So this locking mechanisms or the standards is absolutely essential in the clinical environment. Okay, and in fact that's the reason many of the softwares, what you must have heard, is called CFR Part 11 compliant, which is an FDA code. So that means you cannot tamper with it. Actually has a trail of all the changes. And controls. How do you know that your chemistry actually worked for the clinical samples? We don't have enough controls. And obviously there are a bunch of clinical parameters if you are doing the testing. So these are, you can imagine, I talked about the data challenges, I talked about the biology challenges, I talked about the clinical challenges, then I'm talking about something which is challenges related to the regulation, clinical regulation and standards. Nonetheless, these are also equally important, because keep in mind, this has to be utilitarian in the clinic. And in order for it to be utilitarian in the clinic, you need to have all these puzzles fit together. If you're good in one, you are not that good. You have to be good in all. And on this, so we need to actually come up with standards in the clinical labs, how to do this test, how to do this well, how to do this in the time frame which is acceptable to the patients, and how to do obviously inexpensively without compromising the accuracy. So I just wrote actually an editorial on what happens in genomic diagnostic test, what are the different things that the government and the private players or the academic institutions should do, but this probably doesn't interest you. But just to end, this is my last slide. I'll give you an example which might make sense to you. This is, I don't know how many of you know this car. My son actually loves this car. This is a Bugatti Veyron Super Sports, the most powerful car. Probably cost more than 4 crores. Is 1200 horsepower, obviously pretty fast, and it's a dream machine. He loves it, and this is my favorite car. Now the reason I'm showing you this actually apt expression of what's happening in this field outside in terms of clinical utility and what's happening here. Now this is the problem. This is what happens in the US. Now we need to be here to make it useful for our public in India. Now a test has to be in 5,000 rupees range to be useful for an Indian public. Right now they are in $1,000 and $2,000 to $5,000. Okay. Now I mean, you can read this. It's pretty simple. We need not only to tighten the loose ends at the chemistry side, the biology side, the clinical sides, but also in the regulatory sides, standard side, in making things fit. So the answer to future is working together. We have no choice but to work together in a multidisciplinary way to make sure right from the day one when you get the sample from the clinic to you actually give the answer to the patient. It has to be worked in a very concerted and an atmosphere of congeniality. Thank you.